Welcome to my channel. Today I will discuss about the protein second part. So first let's talk about the secondary structure of the protein alpha helix. Among the alpha helix there are three types like 3.6, 13 helix, 310 helix, and another one is pi helix, what is 4.4 by 16 helix. But most common one is 3.6 13 helix, and also very rare but not very few is 310 helix. Let's talk about 3.6 by 13 helix, what it means. In alpha helix, 3.6 13 helix means here the residue is 3 and another is 0.6 of another residue, and the angle between this is 360 divided by 3.6, that is the 100. That means each amino acid turn 100 degree to complete this structure, 3.6 structure. 360 divided by 100, 3.6 amino acid, and here contain 3 amino acid and 0.6 of another amino acid. I'll show it later. And what about the 13? In each loop, that is the hydrogen bonded loop, we know hydrogen bond built between CO and NH functional group. So in each hydrogen bonded loop the total number of atoms is 13. I will show it later. And in case of 310 helix, again 360 divided by 3, 120. That means each amino acid turn 120 degree to complete this turn, and total number of amino acid residue in one turn 3. And what is the hydrogen bonded loops atoms number? 10. Yes, so here hydrogen bonded loop atoms number 10. Let's see. So here is an example of 3.6 13 helix. I adapted it from the internet, this picture. So let's first talk about the helix occur in right-handed orientation. Why it's right-handed orientation? If we see the pi and psi value, this is an ideal pi and psi value for alpha helix, which is minus 57 and minus 47. Minus means it stay in the third quartile of Ramachandran plot, where alpha helix, right-handed alpha helix are possible to adopt there. So, and another here is minus means they are right-handed alpha helix. What about the hydrogen bonding pattern? If we look closely here, in one first amino acid bind hydrogen bond, this is hydrogen bond with CONH, as I told earlier, is with fifth amino acid. That means one with fifth amino acid, and second amino acid make hydrogen bond with sixth amino acid. And what about third amino acid? Third amino acid, it makes hydrogen bond with seventh amino acid. That means the pattern is I plus 4 patterns of hydrogen bonding. And in each hydrogen bonding, in each turn, there is around 20 to 30 amino acid residue present. What about the piece of that hydrogen alpha helix? Here for 3.6 13 helix, the pitch is 5.4 angstrom. Let's talk about the beta sheet. Generally we found two types of beta sheet. One is parallel beta sheet and another one is anti-parallel beta sheet. If we look closer here, these are anti-parallel in this direction and this one is this direction. What about the piece between these two? Between these two, if we look closer, between these two, piece 7 angstrom. In case of alpha helix, ideally is 5.4 as I told, and in case of beta sheet, around 7 angstrom. And this one is anti-parallel and this one parallel. These two are common. For anti-parallel beta sheet, the ideal pi and psi value is minus 139 and plus 135 for anti-parallel beta sheet, and for parallel beta sheet it's minus 119 and plus 113. That means minus plus. So it must be in the first quartile of Ramachandran plot, where it's fixed for beta sheet. Let's talk about another type of structure which is called super secondary structure. Why super secondary structure? Because it's contain more than one alpha beta sheet, or beta sheet alpha helix, or alpha helix beta sheet, alpha helix beta sheet, or alpha helix with a turn or loop. That's why it's called also motif. I will discuss later about motif and domain. So these are called super secondary structure. Let's talk about the experimental protein design, determination, and also how to predict this protein structure by using computer. Let's talk first about the experimental protein structure. In experimental, we can do it through X-ray crystallography, NMR, and also cryo-electron microscopy. First, let's discuss about how X-ray crystallography used to determine protein in experimentally. Its atomic resolution must be beyond 1 angstrom, and the protein size is not a constant. It's not a problem, but the protein must be crystalline form. If we use X-ray crystallography, it must be in crystalline form. What about NMR, nuclear magnetic resonance? Here is fairly accurate and reveal dynamics, but this one is limited for very small proteins. It's not perfect for larger protein. And cryo-electron microscopy, this is imaging technology, but its resolution is very low. Let's talk about how we can predict protein structure by using computational measure determination of the protein. Here 4 different type of approaches are already introduced so far. First, let's talk about comparative modeling, or its another name is homology modeling. What it means? This actually the protein with similar sequence, that is the one protein which sequence is similar, then we can adopt it with the predicted structure which is similar with this structure. How it's done? First, find a template protein from PDB. PDB stands for protein database, and then it's aligned with the target sequence. It align with the target sequence for that computational model. That means in homology it's comparative. There must be one structure which is known, and then we find which will be sequenced or which will be predicted are similar with this model or with this structure. That's why it's called homology modeling. What about fold recognition or threading? Fold recognition and threading is actually appropriate when a protein sequence has no clear homology in database. In database, when we can't find any similarity of that protein, but that may be adopt a structure similar to known folds, almost similar or closely similar to the database, we can find it. And the popular threading tools are known iTeser, and here comparating or homology modeling more popular, like Swiss model or modeler are very popular for homology modeling. And for fold recognition, as I told, popular iTeser, very popular. And what about initio approach, or another name de novo approach, or from the scratch? That means here we can't find any it's not necessary to take from the database. That is totally newly synthesized from the amino acid. It's build a 3D structure from amino acid sequence without using any template. And most popular, Rosetta and also Quark. But Quark is appropriate for a small protein structure. What about machine learning? Machine learning is AI based and widely used. It uses a vast data set, and the popular machine learning method of protein prediction is AlphaFold and also Rosetta Fold. These are very common for predicting using machine learning. But remember, these are predicted method, and it must be confirmed with the experimentation before using it. Let's talk about secondary structure prediction method. Secondary structure prediction method in first generation, that means in 1970s, single residue statistics was used, like Chow-Fessman method, Gura-1 method, etc., and its accuracy was 50 to 55%. And in case of second generation in 1980s, that is segment statistics, like GOT3, ALB method, and there the accuracy was 60 to 65%. And in case of third generation, 1990s method, these are Cypred, Jpred, and also Gore 4 and 5 recently introduced, but here the accuracy is 70 to 80%. Let's talk about Levinthal paradox. Levinthal paradox is a thought experiment in computational protein prediction. So what it actually means, say for 100 amino acid residue we have to make a protein, we have 100 amino acid residue, so they can form three possible conformation, alpha, beta and gamma per each residue. That means for each amino acid of this 100 amino acid they can form three possible conformation. So total number of possible fold will be 3 to the power 100, that is 5 into 10 to the power 47. That means this 100 amino acid residue can form 5 into 10 to the power 47 possible form conformation. So as we know that time scale of a fold vibration is 10 to the power minus 13 second. What it means? That means to form a new conformation it can take 1 second to form 10 to the power 13 structure. That is the time scale of fold vibration. Time scale of fold vibration is 10 to the power minus 13 second. That means for 1 second they can produce 10 to the power 13 structure for a single conformation. So if we divide this time with the total conformation, that is 5 into 10 to the power 47 divided by 10 to the power 13, then we got the second. How much second? Is 5 into 10 to the power 34 seconds. And if we convert this second into years, it will be 1.6 into 10 to the power 27 years, which is higher than even the universe is. But actually in 37 degrees Celsius 100 amino acid residue can fold within a few seconds or within millisecond even. So how it possible? This is called Levinthal paradox. Let's solve it. To solve this Levinthal paradox, Ken A. Deal and Hu-San Sen in 1997, they told that the protein do not search randomly through all the conformation. Rather, they follow a funnel-like structure, which they called folding funnel hypothesis or energy landscape theory. Let's discuss about this theory. In this theory, first in this one is y-axis is energy from top to bottom, high energy, and then go low energy, and in x-axis this is the conformation. So here this is unfolded condition, wide range, unfolded with the high energy state, and then they go into the pocket, intermediate folding, or sometimes they trapped with this pocket which is called local minima, intermediate energy state. They trap it and form unfolded or sometimes aggregated, which is called misfolded, but most of the time they come here and then go back, then come here, go back, which is called molten globule, that is partially folded protein. And when the energy become minimum, then it comes into here, which you call global minima. Here is the native state, that is the native state we find in global minima, the lower state here at the bottom of this funnel. So this is called folding funnel hypothesis or energy landscape theory or golf course theory. So let's explain it. Here we have got two state. One is local minima. Local minima is intermediate energy state where the protein is partially folded, as I told, partially folded here, and then what happened? But it's not in native conformation. To be native conformation, it must come to the lowest energy state. This is the lowest energy state where it found the native state. Native state structure of the protein found in the lower energy stage, that means in global minima. And then they can be trapped here. They can be trapped in local minima sometimes in non-native conformation, and this sometimes leads to misfold aggregation. And what about global minima? The lowest energy state, which corresponding to native and functional conformation. and this native state or native structure of protein shows the thermodynamically stable structure among all the physiological condition. So if we say again what is actually folding funnel hypothesis, as I told that what Levinthal paradox, Levinthal what actually thought that protein to fold structure a single 100 amino acid residue structure, the protein need even more than universe is, but it's not possible in theoretically because protein structure is not happen randomly. Proteins don't search randomly through all the conformation, rather they are little bit biased to form their native structure. So what they did, they first unfolded condition high energy, then they go through the intermediate condition and they release the energy, and the lowest energy is the native structure which is found in global minima. Let's talk about the protein folding. Protein folding can happen two ways. One is spontaneous protein folding, another one is chaperone-mediated protein folding. Let's first talk about spontaneous protein folding. Spontaneous protein folding, as we know that protein has two type of bond. One is non-covalent bond and another one is covalent bond. What is the non-covalent bond? Like hydrophobic collapse, ionic interaction, hydrogen bonding or van der Waals force. And in case of covalent bonds, disulfide bond between the cysteine residue we have shown. So what about chaperone-mediated protein folding? As we told previously, spontaneous protein folding, that means proteins can fold spontaneously without any help of chaperone, but here chaperone can help to fold the protein. Why chaperone is needed? Because when the self incompatibility of the polypeptide are present there, they can't fold itself can't fold, then they need help to fold. In that case, chaperone help them to fold correctly. And also in some case functional protein damage due to some environmental condition, that is the lost their structure which we call disrupted or denatured. In that case, chaperone can help to correct the folding, to correct the misfolding into the functional structure. Let's talk about the chaperone-mediated protein folding again. They are basically heat shock proteins. Heat shock proteins, they act as molecular chaperone, they helping other proteins to fold correctly. These heat shock proteins help other proteins to fold correctly, and also they help to prevent aggregation and also recover from stress induced damage like heat, drought or toxin. So two type of chaperone found, one is chaperone and another one is chaperonin. Let's talk about this. In chaperone, in eukaryotic it found HSP 70, heat shock 70. Why it's called 70? Because it contains 70 kD kilodalton, and HSP 40 and also HSP 90. These three chaperone found in eukaryotic cell. What about prokaryote? In prokaryote, HSP 70 known as DNA K, HSP 40 known as DNA J. So sometimes you may heard this name DNA K and DNA J. This is just the similar like HSP 70 and 40. But in prokaryote, HSP 90 absent. HSP 90 only found in eukaryote. So in eukaryote, this 70, 40, 90 they help to fold the protein. But here required ATP. Also in case of chaperone, they also need ATP. But here mostly chaperone, it was newly synthesized protein, fresh protein, newly synthesized protein, and that's why sometimes it called they work co-translationally. They co-translationally work, and among these three, HSP 70 is the major chaperone. Let's talk about chaperonin. Chaperonin in eukaryote HSP 60 and HSP 10, and in prokaryotic HSP 60 known as GROEL, HSP 10 known as GROES. That is EL-ES system. Sometimes you have noticed this. So this is the similar like eukaryote HSP 60 and HSP 10. This chaperones look like barrel-like structure, and here the lead-like structure is HSP 10 in eukaryote and GROES in case of prokaryote. I will discuss it later. It also need ATP, and most of the case chaperonin helped when chaperone partially folded, then it goes to chaperonin to complete the folding action. So that means it's not fresh, it's not newly synthesized, it's better older proteins or partially folded or maybe misfolded protein come in chaperonin, and then they complete their folding structure to a functional state. As I told earlier that HSP90 is found only in eukaryote and it's also ATP dependent. It assists in the correct folding a wide range of client protein, and it mostly are available for many signal transduction. In plants also HSP90 involved in developmental regulation, immune response, abiotic stress tolerance like drought, salinity or cold, and HSP90 localized in especially cytosol, chloroplast and mitochondria. Let's talk about chaperone-mediated protein folding again. As we saw, this is our target polypeptide. We have to fold it. That means chaperon will assist to fold this. HSP70 and 90 hold together, and then HSP40 come, like make this structure, and then what happen? Then this complex go into the chaperonin. I showed again this chaperonin they go, and then this within this chaperonin they complete their folding structure. As I told earlier, HSP60 look like barrel like structure, and HSP10 look like lead like structure, and they are polymeric. They are multimeric condition here in both HSP10 and also in HSP60. So here if we look again in this picture, this chaperonin helps to partially folded protein. They enter into here and then lid close, and then when lid open, then correctly folded protein, which is functional protein, came here. So that's how chaperon, molecular chaperon, helps to protein fold correctly. So let's again talk about the roles of heat shock protein in biochemical function. First is facilitate proper protein folding. As I showed earlier, how chaperon and chaperonin complete the correct folding structure, and then they are preventing misfolding and aggregation of protein. They always prevent misfolding and aggregation of protein. And if misfolding occur, then what happen? Then they redirect or they direct them to degradation pathway. I will tell later about the degradation pathway, how ubiquitin and proteasome help, ubiquitin-proteasome system help to degrade this misfolded protein correctly. And the direction, because this misfolded protein need to go ubiquitin-proteasome system to degrade, then who will assist here to direct? Here again chaperon will HSP heat shock proteins will help to direct them to degrade pathway, that is ubiquitin-proteasome system. I'll show it later. And also it helps to stabilize the proteins and membranes. It helps to stabilize the proteins during different environmental stress like heat, drought or oxidative stress. It also maintains overall proteostasis, that is the protein homeostasis, and support help to survive by balancing protein synthesis. That means protein synthesis and also protein degradation, which we call protein turnover. I will discuss it later. Let's talk about protein misfolding. How protein misfolding occur? Protein misfolding happen when protein fails to adopt its correct 3D structure. As I told earlier, that protein in native structure is the correct structure, other one is not correct structure. So sometimes they form misfolding or aggregation of protein, and this misfolding protein can have a toxic aggregation. They sometimes interfere with cellular process. How? Like misfolding, protein misfolding disease is very common, like Alzheimer's in human, Parkinson, and also other disease in mammals. And in case of plants also, Rubisco and photosystem 2 protein sometimes misfold under heat stress. Then what happen? Then carbon dioxide fixation can't occur efficiently. And this misfolding of storage protein also common in different seeds like prolamine in maize due to mutation or stress. So this folding of protein is not uncommon, it's very common. Then how plants survive with this type of misfolding occur? Then how they try to solve this problem? Then they highly rely on chaperones, as I told earlier. If misfold any protein aggregate or misfold, then chaperon helps to direct it towards the ubiquitin and proteosome system, and this ubiquitin and proteosome system will help to degrade this migrate or partially folded protein into amino acids, fresh amino acids. Then this plant or animal system can utilize this amino acid to fold protein again. Let's talk about ubiquitin-proteasome pathway, how plants or animals use this ubiquitin-proteasome pathway to solve their misfolded proteins, to degrade their misfolded proteins. So first, ubiquitination. Ubiquitin itself also a eukaryotic protein, and they are monomeric protein. That means they contain only one polypeptide chain and only 76 amino acid residues they contain in ubiquitin. Let's see about the ubiquitin, how ubiquitin attached with the target protein. Ubiquitin attached with the lysine of the protein, lysine of the target protein. So bear in mind that ubiquitin always attached with the lysine residue of the target protein, and it can occur monomeric, mono-ubiquitinated, and also multi-ubiquitinated also occur in if it occur in different attached in different site of lysine in the target protein. And polyubiquitinated is very common, especially this happen if the cell need to degrade this target. This is the degraded protein. So to be degraded, if for example this one is misfolded, so cell system need to degrade this protein. So the degradation machinery is proteasome. So how it recognize this one need to be degraded? Here is the signal. This signal is polyubiquitin, and it must be more than 4. If it bind with each other in a row and in poly more than 4, then the cell understand that proteasome need to be degraded this type of misfolding protein. But these are not allowed for misfolded. These are uni or maybe multi, but not in a linear way and not in poly. That means always polyubiquitin is targeted to be gone through proteasome to be degraded. Okay. Then how actually ubiquitination process occur? It occur in 3 steps. First, step 1, activation of ubiquitin. One enzyme helps to activate ubiquitin, and then transferred to E1 to E2, E2 E1 to E2. Actually 3 enzymes are involved here, E1, E2 and E3. E1 known as ubiquitin active enzyme. As I told earlier, ubiquitin need to be activated, and to activate ubiquitin this E1 enzyme help, and to transfer from E1 to E1 to E2 enzyme, which is important. E2 help to ubiquitin conjugated enzyme, I will show you later, and E3 ubiquitin ligase enzyme. Okay, then step 3 is ligation of ubiquitin to target protein. Let's see here. These are the ubiquitin. They also utilize ATP to bind. This one the activator. That means E1 help to activate ubiquitin, and then ubiquitin activated ubiquitin attached with E2, and then what happened? E3 help to ligase this E2 into the target protein. This is the target protein, and this is E2, and this is E3. That means E3 enzyme 3 helps to ligate E2 and ubiquitin into the target protein, and then this polyubiquitin with the target protein, which we need to be which is misfolded, and we need to be back them into proteasome to be degraded. Then this enter into the proteasome system. This is the proteasome, again barrel like structure with the lead. I will discuss later. These are the lead. Then this misfolded protein enter into this proteasome, and then with the help of protease enzyme is degraded into small amino acid. These are the fresh small amino acid. That means misfolded protein enter here, and then is degraded into amino acid. That's how plants or animal system use recycling the misfolded or partially folded or aggregated protein. So here again I told earlier that it need to be degraded. Which one need to be degraded? it must have 4 or more than 4 ubiquitin in a row. Then the cell system will understand that it need to be degraded, and it brings into the proteasome system. And if it occur in any side, mono or multi, but not in a row, not in a poly, that not to be degraded. So again, as I told earlier, this is the proteasome. Proteasome, as I told, proteasome has a barrel-like structure which is 20S, and also a cap-like structure or lead-like structure which is 19S in two side, and it's also ATP dependent. That means this one is polyubiquitin, as I told, this one polyubiquitin with the targeted misfolded protein. Then it enter into this 90S cap, and this go through the barrel-like shift, and this is degraded with the help of protease enzyme. Here protease enzyme helps to degrade misfolded protein into the proteasome. Then it degraded it into amino acid residue. So as I told earlier, proteasome is degraded to unneeded or damaged protein. That means it never degrade any protein, functional protein. It's always target non-functional or aggregated or misfolded protein. How it recognize which one is functional, which one is aggregated or misfolded? As I told earlier, it's ubiquitin when polyubiquitin attached inside in a linear way on misfolded protein, then it bring into the ubiquitin proteasome system, and then proteasome with the help of protease enzyme it degrade into the smaller amino acid. That's why it called the cellular recycling center. And as I told, with the help of proteases they degrade the misfolded protein into the smaller amino acid residue, and it present in nucleus and cytoplasm. Let's again discuss about how protein degradation occur. As I told, this one is the protein degraded, misfolded protein. We need to be degraded. It need to be degraded, and then polyubiquitin, as I told, need more than 4 or more than 4 in a tag. These are tagged for degradation 1, 2, 3, 4 in a row. Ubiquitin with the misfolded protein. Then this one is proteasome. Then it enter into the proteasome, and then ATP utilize, and then what happen? Then it go inside here, and then ubiquitin release, and amino acid residue come out. Then again this proteasome go there to use another misfolded protein which is tagged with polyubiquitin. Let's talk about the protein turnover. What is protein turnover? Protein turnover, this is a continuous process of protein synthesis, that is the creation of protein, and protein degradation, that is the breakdown of protein. And why it's important? It's important to maintain cellular protein homeostasis, that we call proteostasis. So how it's occurred? Our body or in plant part also protein synthesis occurred through the amino acid pool, and then it produce the protein, and proteolysis, different proteolytic activities, protease or others degraded this amino acid, and then this amino acid again stay in the amino acid pool. How this amino acid pool come? From the dietary amino acid or from the proteolysis of the protein, and then this amino acid catabolism occurred and from urea and carbon dioxide. This is another story. And then this biosynthesis of nitrogen compound, from this amino acid pool some other biosynthesis of nitrogen compound occur. This is another story. And so protein synthesis, how protein synthesis occur? In ribosome translate mRNA to build new protein. And how protein degradation occur? Ubiquitin-proteasome system like autophagy, lysosome system, or other protease like CLP protease, which is also common in plants. This is all about protein turnover. This is a continuous process of protein synthesis and protein degradation. Let's go about the summary. So most of common alpha helix type is 3.6 by 13 helix, which means in each turn it contains 3 amino acid and 0.6 of another amino acid, and the angle between the amino acid is each amino acid 100, and they contain 13 atom in each hydrogen bonded loop, and the Helix piece is 5.4 angstrom, and the ideal phi and psi angle is -57 and -47. That means it must stay in the third quartile, where minus portion find of Ramachandran plot. And in case of beta sheet, two type of beta sheet present, antiparallel and parallel. And antiparallel, its ideal antiparallel pi and psi angle will be -139 to +135, that is minus plus. It must come in the first quartile of Ramachandran plot. And also in parallel beta sheet, same, it's ideal for parallel -119 and +113 degree, and the piece distance is 7 angstrom. About the Levinthal paradox. Levinthal paradox is a thought experiment, as I told earlier, that if we think that 100 residue they can make 100 amino acid residue, they can form in 3 different way to establish any structure, alpha, beta and gamma, as I told. So their time scale of fold vibration is, if 10 to the power 13 second, then it will take 1.6 into 10 to the power 27 years to fold, which is quite impossible, while a 100 amino acid residue can fold within milli or few seconds. So this is paradox, and this paradox solved by the one hypothesis, non-folding funnel hypothesis, when it's so told that it has actually 3 structure. First, in the top of the funnel unfolded, then intermediate molten globule, which is semi or partially folded, and also the native structure, which is the lowest free energy state. That is, the lowest free energy state is the global minima, which is the native structure, and again local minima and global minima. Local is intermediate energy state, and global minima is the lowest energy state. So, and this native structure is the thermodynamically stable structure of any protein. And what about protein folding? Protein folding can occur 2 way, spontaneously or chaperone-mediated protein. And spontaneously, how it occurs? As we know, it has a lot of covalent and non-covalent bonds, so they help to fold spontaneously, naturally, without any help of chaperone. And also chaperone can help to fold the protein correctly. Chaperon has 2 types, chaperon and chaperonin. They both helps to mediate protein folding. And chaperon is HSP 70, 40, 90. These 3 works together to fold protein correctly. And also chaperonin, which is HSP 60 and 10. HSP 60 is barrel-like structure, and HSP 10 is lead-like structure. This both HSP 60 and 10, they act as the chaperonin. Here chaperon helps to synthesize, to fold newly protein, that is the fresh protein, and chaperonin partially folded or misfolded or sometimes older protein, they help to fold. And what is ubiquitin-proteasome pathway? As if any misfolded protein occur, it's very common, so many misfolded protein can occur, as I told earlier. So this misfolded protein can be degraded into fresh amino acid through a system. This system known as ubiquitin-proteasome system. To be degraded misfolded protein, it must be tagged with ubiquitin in polytag, not one or two in different part will not be effective. It will not give any signal to the proteasome that it need to be degraded. To be degraded, it must be polyubiquitin, 4 or more than 4. This ubiquitin attached with the lysine residue of the targeted misfolded protein, and polyubiquitin, as I told, 4 or more, transfer them to the proteasome, and then proteasome degraded into amino acid, which was done by the enzyme of proteases. That's all about today's lecture. Thank you for watching.